How ancient humans used toxic plants to create the first medical treatments. You are standing beneath the bright white lights of a modern pharmacy. Your head hurts. Not catastrophically, not heroically. It is the kind of headache created by 8 hours of screens, bad posture, too little water, and one meeting that should have been an email. You pick up a box of pain relievers. Then you turn it over. The warning label mentions stomach irritation, drowsiness, allergic reactions, interactions with other drugs, and several conditions that sound serious enough to make you briefly reconsider whether the headache is actually that bad. You are holding a measured dose, manufactured in a regulated facility. The active ingredient is known. The tablet is sealed. The instructions are printed. And you are still suspicious. Now remove the pharmacy. Remove the label. Remove the doctor. Remove the laboratory that tested the compound. Travel back tens of thousands of years. Your jaw is swollen from an infected tooth. Your skin is hot with fever. Every heartbeat sends pain through the side of your face. Around you is a forest containing thousands of chemical compounds. Some can reduce inflammation. Some can repel insects. Some can slow the heart. Some can stop the heart. The same plant may help at one dose and kill at another. There is no measuring spoon. There is no poison control center. There is only memory, observation, experience, and the terrifying possibility that the person teaching you is wrong. So how did ancient humans learn to turn a landscape full of plant defenses into the first medical treatments? The answer is not that one brave genius randomly ate every leaf until something worked. That strategy produces a very short medical career. The real answer involves animal behavior, bitter taste, controlled preparation, social learning, repeated observation, and knowledge passed through generations. It also involves uncertainty. Archaeologists can identify plant residues. They can identify disease. They can sometimes connect the two. But a trace of a medicinal plant is not a written prescription. The first pharmacy left no instruction manual. It left clues inside teeth, bedding tools, fire residues, and human bones. And one of the most revealing clues came from something most modern people pay to have removed: dental calculus. Plaque can harden onto teeth and trap microscopic pieces of a person's life. Plant fibers, starch grains, smoke compounds, microbial DNA. Tiny chemical traces can remain locked inside that mineral layer for tens of thousands of years. In 2012, researchers examined dental calculus from Neanderthals at El Cedrón Cave in northern Spain. They found evidence consistent with cooked plant foods. They also detected compounds associated with yarrow and chamomile. Those plants are interesting because they are bitter and provide little obvious energy compared with meat nuts or starchy plants. Today, both are associated with medicinal uses. Yarrow has a long history in wound care and preparations intended to manage inflammation. Chamomile has traditionally been used for digestive discomfort and spasms. Did the Neanderthals at El Cedrón use them as medicine? Possibly. The combination of bitterness, low nutritional value, and known bioactive properties makes that interpretation reasonable. But reasonable is not the same as proven. The plants may have been used as flavoring. They may have entered the mouth through food preparation. Someone else may have prepared them for a sick individual. Or they may have served a purpose we have not considered. This distinction matters. Ancient people were knowledgeable. That does not give archaeologists permission to turn every bitter leaf into a prescription. 5 years later, the story became even more striking. Researchers sequenced ancient DNA preserved in Neanderthal dental calculus. One El Cedrón individual had evidence of a dental abscess. The same individual also carried a gastrointestinal pathogen. Inside the calculus, researchers identified poplar DNA. Poplar contains salicylate compounds related to the chemical history that eventually led to modern aspirin. The researchers also detected DNA from Penicillium mold. That combination created an irresistible headline. A sick Neanderthal had taken a natural painkiller and an antibiotic. It is a fascinating possibility. It is not a prehistoric medical chart. Poplar may have been chewed for pain. It may also have been consumed for another reason. Penicillium includes many species. Its presence does not prove that the mold produced a therapeutic quantity of penicillin. It does not prove intentional treatment. And it does not mean a Neanderthal invented combination drug therapy 50,000 years before germ theory. What it does show is that this individual was sick and had contact with biologically active, natural materials. That is enough to open the door. It is not enough to pretend we can see the entire room. The strongest evidence for ancient plant knowledge does not come from one dramatic tooth. It comes from patterns. Plants were selected. They were processed. They were placed in living spaces. They were heated. They were carried. And in some cases, they were chosen from many available species for properties unrelated to food. At Sibudu Rock Shelter in South Africa, people were constructing plant bedding about 77,000 years ago. The bedding included sedges and other plants. Its upper layer contained aromatic leaves from Cryptocarya woodii. Those leaves contained chemicals with insecticidal and larvicidal properties. The people at Sibudu lived near a river. Aromatic insect-repelling leaves would have been valuable in a place where biting insects could disturb sleep and transmit disease. Beginning several thousand years later, the bedding was also burned. The most cautious interpretation is site maintenance. Burning old plant material can remove waste and pests and prepare the living area for a fresh layer. That is not a hospital. It is preventive environmental management. And prevention may be one of the oldest forms of medicine. You do not need to understand microorganisms to notice that people become ill less often when sleeping areas are cleaned. You do not need to identify an insect species to notice that one leaf keeps biting creatures away. You do not need molecular chemistry to recognize a reliable pattern. Use this plant. Avoid that one. Burn the old bedding. Do not drink from that pool. Apply this preparation to that kind of wound. Stop if the patient begins shaking. The knowledge begins as observation. Then it becomes procedure. Then it becomes tradition. Now imagine a healer in a prehistoric camp, not a mystical wizard surrounded by glowing bottles. A practical observer surrounded by people who remember what happened last time. A hunter has returned with a swollen jaw. He cannot chew. He is feverish. The group knows that the condition can become deadly. They also know that chewing bark from a particular tree sometimes reduces pain. No one knows about cyclooxygenase enzymes. No one knows about inflammatory pathways. They know something more immediate. The bitter bark helped one person. Too much made another person vomit. A different tree did nothing. Fresh bark behaved differently from old bark. Heating changed the taste. Children responded differently from adults. The treatment is not discovered in one heroic moment. It is refined through accumulated cases. This is the essential technology, not the plant by itself, the memory around the plant. A poisonous landscape becomes medically useful only when information is attached to it. Which species? Which part? Which season? Which preparation? Who should receive it? What signs mean the treatment must stop? Those questions are pharmacology before pharmacology had a name. But this knowledge came with a brutal cost. Plant chemistry varies. Two plants that look similar may contain completely different compounds. The concentration can change with soil season, rainfall, age, and plant part. A dose that helps one person may harm another. Some effects take hours. Others take days. A patient may recover because of the treatment. The patient may recover despite the treatment. And if someone dies, the group must decide whether the plant failed, the dose was wrong, the illness was too severe, or the treatment caused the death. Without controlled trials, those possibilities are difficult to separate. This is why ancient medicine should not be romanticized. It contained genuine knowledge. It also contained dangerous mistakes. Effective remedies could exist besides superstition. A useful preparation could be combined with an ineffective ritual. A healer might save one patient and poison the next. Nature is not automatically safe. Many plant compounds exist because plants are fighting not to be eaten. Alkaloids can interfere with the nervous system. Cardiac glycosides can alter heart function. Some compounds damage the liver. Others affect blood clotting, muscle control, or consciousness. The principle later summarized as the dose makes the poison is real. But prehistoric dosing was not a magical act of perfect calculation. It was a difficult cultural achievement built from repeated experience. And we should not turn that reality into a dangerous recipe. The archaeological record does not tell us an exact dose of nightshade foxglove or any other highly toxic plant used safely in deep prehistory. Inventing one would not be scientific. It would also be reckless. What we can reconstruct is the logic. Ancient humans learned that preparation changed effects. Crushing releases compounds. Heating may reduce some toxins and concentrate others. Soaking can remove soluble chemicals. Combining materials can alter taste or action. Applying a substance to the skin is different from swallowing it. Smoke affects a living space differently from a drink. This was experimental knowledge. And experiments do not always require formal laboratories. A laboratory is a system for making observations repeatable. A prehistoric camp could do that through shared routines. The group may also have learned from other animals. Scientists call animal self-medication zoopharmacognosy. Wild chimpanzees sometimes swallow rough leaves whole. They do not chew them like ordinary food. The leaves pass through the digestive system and can help expel intestinal parasites mechanically. Chimpanzees also chew bitter pith from certain plants during episodes associated with parasite infection. These behaviors do not prove that early humans copied a specific chimpanzee. They show that selecting unusual plant materials during illness is older than written medicine and may have deep evolutionary roots. An attentive human could watch animals closely. Which leaves do sick animals seek? Which bark do they chew only during certain seasons? Which plants do insects avoid? Where does an injured animal rest? Observation provides candidates. Human culture tests, remembers, modifies, and teaches. That final step changes everything. An animal may discover a useful behavior. Humans can explain it to someone who was not present. They can carry a plant to a new place. They can preserve it. They can compare outcomes across many patients. They can warn children about look-alike species. They can build a medical tradition larger than one individual memory. This is why the first medicine was probably not invented by one person. It emerged from a network. Hunters knew animal behavior. Gatherers knew seasonal plants. Older people remembered rare reactions. Caregivers watched symptoms over time. Toolmakers created grinding stones, containers, and cutting edges. Fire users transformed raw materials. Language joined those observations into instructions. The medicine was chemical. The medical system was social. As humans moved into new environments, the library expanded. Different regions offered different compounds. Willow and poplar contained salicylates. Opium poppies produced alkaloids that could suppress pain and also stop breathing. Foxglove contained cardiac glycosides that later contributed to heart medicines, but could be lethal in uncontrolled amounts. Cinchona bark provided quinine-related compounds. Sweet wormwood eventually yielded artemisinin. Curare mixtures used by some indigenous South American societies contained muscle-paralyzing compounds that later helped inspire controlled medical muscle relaxants. These discoveries did not all occur in the Paleolithic. They came from different peoples at different times. They also did not move directly from leaf to modern tablet. Traditional use provided observations and chemical leads. Modern science then isolated compounds. It measured doses. It tested toxicity. It compared treated patients with controls. It identified interactions. It rejected many remedies that did not work. It modified molecules. It standardized production. That is the real relationship between ancient and modern medicine. Modern pharmacology did not simply replace plant knowledge. It turned uncertain natural variation into testable, reproducible treatment. Consider aspirin. Its history is connected to salicylate-containing plants, such as willow. But the tablet in your hand is not powdered prehistoric bark. It is a defined chemical dose with known benefits and known risks. Consider morphine. Its origin lies in the opium poppy. But modern medicine does not treat every poppy preparation as equivalent. Purification and measurement make the effect more predictable. Consider artemisinin. The compound came from Artemisia annua and from knowledge preserved in traditional Chinese medical texts. Its successful development required historical scholarship, extraction experiments, chemical analysis, clinical testing, and standardized manufacturing. The plant was the beginning of the investigation. It was not the completed medicine. This creates the final twist. Ancient healers were not primitive pharmacists who already possessed every modern answer. Modern scientists are not detached inventors who created medicine from nothing. They are participants in the same long human project. Observe suffering. Search the environment. Identify a pattern. Share the result. Test it again. Reduce the harm. Improve the dose. Discard what fails. Preserve what works. Your visit to the pharmacy is the newest layer of that process. You pay for the pain reliever. You step through the automatic doors. Outside the city is glowing. Cars move under electric lights. The warning leaflet is still folded inside the box. For a moment, you look at the tablet and imagine the distance behind it. A sick Neanderthal chewing plant material in a cave. A community at Subudu placing aromatic leaves across the floor. Generations watching animals, plants, wounds, fevers, and recovery. Healers remembering which bitter substances helped and which ones killed. Chemists isolating molecules. Doctors testing treatments. Factories producing identical doses. Regulators printing warnings in letters almost too small to read. The tablet is not proof that our ancestors understood molecular biology. It is proof that human beings have been building knowledge from nature for an extraordinary length of time. And the warning label is not a symbol of modern weakness. It is the achievement ancient medicine could not provide. A record of the risk. A measured dose. A method for knowing when not to take it. Ancient humans began medicine by entering a chemical minefield and remembering the paths that did not kill them. Modern medicine inherited that map. Then it added measurements, experiments, and rules. So when you swallow one carefully manufactured tablet with a glass of water, you are not abandoning the world of prehistoric healers. You are using the latest version of their most important invention. Not a particular plant. Not a particular poison. The invention was the idea that nature could be observed, tested, remembered, and transformed into care. That was the first medical treatment. And it may be one of the reasons our fragile species survived long enough to build a pharmacy at all.